Hanna will tell us about plastic in-plane nanopore sensors for biomedical applications.
Hi, everyone. So just building onto the foundation of Maggie's talk about the advantages of plastics and their versatility, I will now address in-plane nanopore sensors for biomedical applications. So what exactly do we mean by in-plane nanopore sensors? There are two primary classes of nanopores. There's the biological nanopores, which are pores made out of proteins embedded in a lipid membrane. A classic example that are commonly used in research is the alpha hemolysin. The first DNA translocation experiments were performed with an alpha hemolysin pore. It has a diameter of about 1.4 nanometers, so it's relatively small. In fact, all biological nanopores have a size range of roughly less than 10 nanometers or so. And so the small diameter allows detection of a limited range of analytes, including ions, small molecules, RNA, DNA, and small proteins. An advantage of these biological nanopores is that they have high reproducibility, right? Because when you produce these proteins, if they were produced correctly, then that should have the same folding and conformations. And so they're highly reproducible in size and shape. But that also leads into their disadvantages is that they're limited size. There's no way you can really manufacture them with a different size using the same alpha hemolysin. And because they are proteins, they also have low chemical stability. So, they're pretty unstable in conditions such as high pH, high salt, buffer conditions. So then comes the solid state nanopores, which are nanopores fabricated in synthetic membranes such as silicon, glass, or polymers, just to name a few. Advancements in nanofabrication techniques allow us to fabricate pore sizes ranging from 1 to hundreds of nanometers. And so with such a wide size range, it allows us to detect bigger molecules such as viruses, nanoparticles, extracellular particles, and even colloidal particles. An advantage of these solid state nanopores is their higher chemical stability as well as fabrication flexibility and a lot more choices of substrate materials. Within the solid state regime is the outer plane nanopores, which are pores that are oriented in such a way that the direction of the translocation is perpendicular to the membrane of the material. And then there's the in-plane nanopores, in which the pore is oriented in such a way that the direction of the translocation is parallel to the membrane substrate. This actually offers a lot more flexibility in designing the architecture of the sensor than the outer plane nanopores. So as I mentioned, with the outer plane nanopore sensors, besides being able to modify the pore size and shape, there's not much flexibility. It's just a membrane with a pore inside a fluidic chamber. There's not much you can design to change the architecture of the outer plane nanopore sensors. But with the in-plane nanopore sensors, you can modify the pore, channel size, shape, add on multiple pores in series, multiple pores in parallel. You can also integrate it into a larger fluidic network for upstream processing, such as purification or surface modifications prior to detecting these analytes. This image here shows the sensor architecture that you will be using in this workshop. It has two different microchannels. The first one filled with a red colored dye, the second one filled with a blue colored dye. These two microchannels flank the nano region. And in the nano region, it can contain a single nanopore, multiple nanopores in series, or multiple nanopores in parallel. So as you can see here, two nanopores with a nanochannel connecting the pores, and then these have 5 pores in parallel. And you can modify the pore size and geometry as well as the architecture to fit the needs of your application. one such example is that, you have virus particles that's different size, different shape. These particles behave differently in resistible sensing than, say, proteins, which can adopt different orientations, different conformations as they go through the pore, which behave differently than, say, flexible polymers like DNA, which also behave differently than small molecules like amino acids or ribomononucleotides. So another factor to consider is the barrier to entry. And all analytes experience a barrier for entry to some extent. And an example I like to use is the translocation of DNA molecules. So DNA is highly flexible, right? In its natural state, it's gonna coil into a ball. So in order to translocate through the pore, it needs to uncoil and linearize itself enough so that it can thread through the pore. You can increase the voltage to help accomplish this, but increasing the voltage also increases the velocity of the analyte. And so depending on your system setup, the analyte might translocate through too fast through the pore for your electronic system to detect, or it might translocate so fast that it's gonna cause signal distortion. So in some situations, it might not always be ideal to increase the voltage. And so one thing you can do to alleviate this issue is to design and integrate an inlet structure to provide a smooth transition from the microchannel to the nanochannel. So Dr. Sunguk Park's group actually investigated this. They designed a nanosensor with 5 different inlet structures. One's a pillar shape that's kind of funnel shape with pillars in the inside the funnel to help uncoil the DNA. And then the actual funnel shape with a wider entrance to provide a smoother transition from the micro to nano structures. And then there's the thin groove, and then the original design where it's just a blunt interface between the micro to nano channel. And then there's the V groove with 2 different entrances. This is a video of labeled DNA translocations. And as you can see over time, you can see translocations of the DNA through this inlet structures. And I think the paper mentioned that out of 300 translocations, only 2 of them actually went through the original inlet structure design because the blunt face just doesn't provide a smooth transition from the micro to nano regime. So the cream of the crop of this experiment is the funnel due to its wider entrance from the micro to nano structure. So it provides a much better sampling efficiency compared to the other inlet structure designs. So now that you know what in-plane nanopore sensors are and their advantages, why plastics? As Maggie mentioned earlier, there's a wide selection of substrates from which to choose to meet the application needs. The surface modification methods are well studied. And then this is a power spectral density, which shows the noise level at different frequencies of the data that was collected. And I believe Mr. Colin McKinney will talk about signal noise in RPS later on, but I just want to mention that plastics provide a much lower noise level compared to other materials such as silicon or glass. And then, of course, plastics are amenable to mass production via injection molding, and they have a simpler fabrication process overall. So Maggie also mentioned thermal fusion bonding with these nanofluidic sensors. They're hybrid devices, meaning that the substrate is made out of a different material than the cover plate. This is an example of a PMMA/COC8007 device in which the PMMA has a much higher TG than the cover plate. So during the bonding process, imagine it, so this is, the side view of a micro fluidic device, but imagine if these are nano features. They're very highly sensitive to deformations during the bonding process. So if you heat up the substrate to their TG, they're going to deform under the bonding process. So you want to maintain the substrate at a solid phase during the bonding process, but you want to heat up the cover plate to their glass transition temperature to help seal, to cross-link the polymers and seal it with a substrate while maintaining the structural integrity of the nanostructures underneath. So the bonding process involves three different variables. There's the bonding time, bonding pressure, as well as the bonding temperature. So to carefully optimize the thermal fusion bonding of your nanofluidic device, you need to carefully control these variables. Too low of a temperature and you risk delaminating the cover plate, but too high of a temperature and you risk sagging of the cover plate into the channels, which obstructs fluid flow. Too high of a bonding time can also sags the cover plate, as well as too high of a bonding pressure. But an advantage of thermal fusion bonding of plastic devices is that you can modulate the pore size during the bonding process. So keeping bonding temperature and bonding time constant, you can modify the bonding pressure to modulate the pore size. Here's an example of a device that was bonded at 110 psi, and as you can see, the pore size is at 22 nanometer after bonding at 110, but when bonded at 170 psi, it decreased to about 12.6 nanometers. And these graphs show, the impact of bonding temperature on the depth of the nanopore as well as the cross-sectional area. Higher bonding pressures leads to smaller pores with smaller cross-sectional areas. And modulating the size helps you improve the signal to noise ratio of your method. Since resistive pulse sensing involves the displacement of ions during the translocation, smaller pores will have smaller probe volume. So if you have a bigger analyte going through, it's going to generate a much higher signal than having the same analyte going through a much bigger pore. So bonding at high temperature here leads to higher event depth than bonding at lower pressure. Maggie also talked about surface modifications. So with DNA, they're highly negatively charged. So if you have a surface that's also highly negatively charged, it's going to repel the DNA, making it harder for the DNA to go through the pore. So you want to cap that with a neutral molecule that also allows hydrophilic surface, but neutralizes the negatively charged surface that's generated during the UV ozone or oxygen plasma activation. So here you can see the surface modification of tris molecule to the carboxylated groups on COP surfaces, capping the negatively charged, but still generating the hydrophilicity of the surface for fluid flow. An application that we're currently looking into in our group is the identification of nucleotide monophosphates for the RNA sequencing. So there are 4 different ribonucleotide monophosphates, each with slightly different structures. And as such, they confer different electrophoretic mobilities. And so in this design, we have 2 pores in series with pore dimensions by 10 by 10 by 12, width by depth by length. As I mentioned, different ribonucleotide monophosphates have different electrophoretic mobility. So by generating a database of those different characteristics, such as the time that it takes to go from pore 1 to pore 2, their dwell time, their peak amplitude, we can generate a database of resistive pulse sensing signals for each analyte towards the ultimate goal of RNA sequencing. Another application is the identification of full and empty adeno-associated viruses, or AAVs. So these are being used in gene therapy. AAVs are composed of a protein capsid shell. Full AAVs contain a DNA molecule that's being used for therapeutic. While anti-AAVs do not contain those DNA molecules. So during the manufacturing of these AAVs, there's a high percentage of the presence of anti-AAVs, which increases the dosage that's required to achieve the same therapeutic effect. So you want to have a method that can detect the ratio of these full and anti-AAVs to help optimize the production process of these AAVs. So in this application, we have the same sensor design with 2 pores in series with slightly bigger pore dimensions to detect the much bigger AAV particles. Same thing, we detect their time of flight, which is different because the DNA confers a slightly different charge relative to the anti-AAVs. Another application is the monitoring of hepatitis B viral capsids during the capsid assembly process. So these capsids are also made out of a protein capsid shell, and these are done with without the DNA in the middle. So it's important to study capsid assembly because then you can target drug candidates that could target certain structures on the capsid to disrupt their assembly for drug development. In this application, 8 pores in series were being used. And with the higher number of pores, it generates better precision. So as you can see from this graph here, 8 pores allows you to make 8 measurements, decreasing the percent RSD of the measurement. And this actually allowed them to differentiate between the time of flight of 2 different capsids, the T3 and T4 capsids, which supposedly have similar charge density, so they should have similar electrophoretic mobilities, but their improved measurement allowed them to detect a slight difference between the electrophoretic mobility for the T3 and T4. And this is an example of the data trace. And as you can see, there are 8 pulses in series representing the 8 pores in series. And here, the improved precision allowed them to detect not only the T3 and T4, but also the intermediate assembly products. And this was actually done by the Jacobson research group. They use a glass substrate and cover plate, but I wanted to include this to show that, you can use more than just 2 pores in series for your application. So another one that our group is focusing on is isolation and enumeration of viral particles in extracellular vesicles for disease diagnostic and monitoring. So this was the device with 5 pores in parallel, which increases the measurement efficiency and reduce the measurement time. So with this device, they were able to lower the limit of detection to 5.5 times 10 to the 3rd particles per mil, which was an 1100-fold improvement compared to an earlier experiment using 1 in-plane pore device. I think that was it. Do you guys have any questions?
Anyone? Hey, if not, then thank you so much for presenting.